Okay, folks, Mr. B here. Today's lesson will be about cell theory with a dash of where the heck does life come from? So let's get to it. Where do living things come from? Well, for the longest time, when our little friends, the maggots here, would show up, it was as if they came from nowhere. They'd show up in spoiled food, rotting meat. It's like, what's going on? And you can only know what you can know, and you can only observe what you can. Humans don't have the best observational ability. We don't see really small things that well. We have a limited hearing range. We can't see except for the colored light spectrum, red, orange, yellow, blue, green, indigo, and violet. We have a very limited range. And so we're quite easily fooled. Magicians have learned these tricks quite well. So this idea, like, where did this come from? We were left to our own devices. We didn't know what to think, right? Until later on, we were able to see microscopic life. Okay, the problem was they had this idea called spontaneous generation. Like spontaneous, like it happens automatically, and generation of life. And the thing about spontaneous generation of life is that's not what happens. Okay, so this is a mistaken concept, so I'm going to get out the big red pen of doom and say that's not what's going on. Okay? When we were able to observe, right, what was happening, flies lay tiny little eggs, right? Sort of think about this. When they're cruising around your house, you leave out food, you're like, are they dropping these things, right? That then grow into a full-featured larval form of the fly before they again start flying around and laying their eggs in our stuff, right? These little tiny eggs, almost microscopic in a way because you can't see them, right? Tiny, tiny eggs, right? So the eggs led to the larva, which you see here, until eventually you get to flies that fly away. Is that why they call them flies? I suppose so. So life doesn't come from non-life. That was a misunderstanding for a long period of time, right? Spontaneous generation and rotting meat. No. So there was this mistaken idea. And once we applied science and we went with our observations, we're able to determine what was going on. Here's a good example of some of the work that had been done. Okay, so I want to start with this. It's also called the Needham experiment. This has also been an experiment referred to as the Spallanzani experiment. That's not important. What is important is if you have, let's say, a nutrient broth, right? Lots of yummy goodness in it. If you heat it up, which is what we do, right, to kill off the germs, because we know about microbes. You want to cook your food to get rid of things that shouldn't be there. Well, if you left it uncovered, like you could see in the above example, and you leave it exposed to the air, there's all sorts of stuff that can get in there. The bacteria can ride in on dust, and so can mold, and eventually, uncovered, you just asked for living cells to make their way back in, whether or not you could see them, and yikes, you get a nice murky flask, which I wouldn't recommend consuming. But if this experiment was done again, where it was sealed. Now, I wouldn't recommend sealing it while you're heating it like that, but I would seal it afterwards. And then if you sealed it to prevent things from getting in there, right, like the germs and the microbes that you can't see that are riding down in air and dust and in the, literally all around us, we can't see them, freaks me out, but that's okay. That broth would stay clear because there's no cellular life in there to grow and divide and take advantage of all these nutrients. So when you look at the Spallanzani experiment, the Needham experiment, largely it was the gravy thing, right? Heat up the gravy, leave one flask open, leave one sealed. And then if the one that's open just starts getting grody, whereas the one that's sealed is free from microorganisms. So you sort of think about processes nowadays where we do even better than this. We don't just heat up stuff, right? We use things that make it hard for microbial life to survive. Think about jams and pickling and all that kinds of stuff. Yeah, we kind of do other things. But an experiment like this proves that spontaneous generation, well, it can't, if life was just going to pop up out of nowhere, why didn't it show up in the one that was covered? because life had to come from life. That's why. Oops, sneaking ahead of myself here. So, spontaneous generation disproved, right? It was an idea. It was an idea. We know that living things develop from other living things. So, for example, looking at these cells right here, right? Our little friends here start as a single cell. And when they clone themselves, they'll make 2 daughter cells. There it is, the nucleus with the little dark spot in the middle. Looks like a grape seed. I always love that. Right, there we go. Another one. And then these can give rise to 2 more cells, like that guy, or maybe that one. And this one could turn into these 2, right? Right, maybe that's where they came from. And literally, when you look, you start from just one itty bitty cell. Then over time, the population just goes up, right? Exponential growth. So you'd be looking, if I was drawing a graph, which I'm apparently not, right? If you were looking at time, independent variable, right? And the number of cells, drawn very crudely, right? If you keep dividing by 2 like this each time, if you go 1 to 2 to 4 to 8 to 16 to 32, you get the idea. It starts to get really add up after a while, right? Holy smokes, that's a lot of cells. And bacteria can do this in 20 minutes. It's very fast. So don't leave your food out uncovered, kids. That's not a good thing. So cells give rise to cells, right? There is no spontaneous generation. That's not a thing. Now, if we kind of go back through history, sort of say, okay, right? We were looking at the simple microscope, right? And we were looking at Anton von Leeuwenhoek's microscope and Hooke's microscope, right? Leeuwenhoek had the one that kind of looked like a magnifying glass, so to speak. Once microscopes really became more of a thing, especially once they were compound with 2 lenses, then we could really see what was going on. In the 1660s, or let's say the middle 1600s, Hooke was looking at cork. That's a material you'll find underneath the bark. And he saw these things, and he called them cells. So he's looking at all these little, right? I'm drawing them much bigger than they are. These little, they seem to line up like this. Very ordered, right? He was wondering, what the heck is going on? Like, there's this stuff inside a plant that's super well ordered. And he termed them cells, but we have to sort of look at why. What's up? Where did that come from? Well, the term for cells, right here, comes from the cork he was looking at. So here's some of the early drawings, this little charcoal kind of sketch he did, sort of pencil sketch. And when he looked at those things, he thought of a monastic cell, like in a monastery, right? And I had a student say to me one time, it looks like a jail cell. Well, it was meant to be pretty simple. You didn't need a lot of things. You needed some light to read by, and you needed, a place to eat and a place to read, and you needed a bed to sleep. And it wasn't exactly like there was carpeting, and quite well ventilated apparently. So yeah, when he looked at all these monastical cells and just how many would be in a monastery, right? He said, well, these little spaces look like cells, right? Not like jail cells. Rather, they're talking about monastical cells. So that's where the term cell came from, right? Like in a monastery. And the name stuck. It's been the name ever since, right? Cool. So the microscopes he was looking at, actually that warrants an investigation. So you're busy writing down like a monastery or reading it or thinking it's awesome. By the way, remember Hooke is spelled with an E. Everybody forgets that. Let's go back here for a second. That's smaller. Okay. Now, Hooke's microscope. Oh yeah, this is what I wanted to show you. So it was kind of interesting, right? Where you had a light source, and in some cases, you had something that would concentrate the light source down on to the stage of what the light microscope was looking at. You could sort of see using something as like a lens, like a condenser, just so that the light would focus on the object on the stage, right? It's a very simple microscope. It works. You could have an oil lamp on one side if that's what you wanted, but you do have to condense the light. You got to pull it in, right, on what you are looking at. Sure, there's an objective lens and an ocular lens there, but you sort of think about it, this is actually pretty slick looking equipment for the time. I mean, that's nice. It doesn't do as well as what we have in the lab, but it'll do. It'll do. So cool looking stuff, cool beans. Some of the earliest drawings, I particularly like this one, where you've got the meandering of the critter kind of drawn like this, right? And looking under the microscope, there's a great number of things you could see. You could see our pond water friends, right? Bits of plant material. Really, if you want to see what you could observe in just a drop of water, let's add this on, right? Let's just say one group, which we study in grade 11, right? All you have to say is protists in water, and I'll just zoom to images, and you can see all kinds of stuff, like the microorganisms in a drop of water through a microscope, right? You got plant-like ones, animal-like ones that look like fungus, right? The one with the tentacles is pretty cool there, kind of in the black and white in the middle bottom there. That's called Hydra. Gotta love that. Like freshwater kind of jellyfish looking thing. But we started to see all this life teeming around, right? As our lenses got better and better, we could see more and more things. But Robert Hooke was definitely getting by. So was Anton van Leeuwenhoek. It was an entire microscopic world that nobody had seen. It hadn't been described. It was cool, right? You see plant material, like, wow, that's amazing what you could see. It's an entire world just underneath your notice. And like amoebas. I always love amoebas, right? Cruising around and swirling and eating things. But don't worry, we'll look at those. We already kind of have in class. White blood cells are very amoeba-like. So early drawings. But it really comes down to the cell theory, which was topic 1.1, right? So based on the work of what they were seeing under the microscope, they said, hey, right? Once spontaneous generation was disproven, they're like cells come from cells. So you've got one cell and it becomes two, and this really should show another set like this, There we go. How's that for 4? Because I did say it goes 1, 2, 4, 8, 16, and so on, right? So from what we started to observe, we noticed that cellular life was giving rise to cellular life. So your cheek cells give rise to more cheek cells, right? All these cells came from pre-existing cells. Makes sense. Our little friend down here, our little friend the amoeba, is just one cell. It's actually cool. It can pull off his whole life. His whole life is just one thing. Kind of like being a white blood cell forever. But it can swirl out. It can move out these little extensions of its cytoplasm. It's like it could push its skin out, and then it can move around things and swallow and eat this poor little algal cell. It's just gonna pull it in and eat it. So the cell theory explains this. There's no spontaneous generation. Cells are coming from cells, right? That's the idea. Cell theory. 3 main pillars of the cell theory. Cells come from cells, okay? So, it's the idea. Cells come from cells here. Then we'll back up and say every living thing is made up of cells. So we'll go here now. Going in reverse. So even if you look at an amoeba, which looks a lot like your white blood cells, and then they can stream their cytoplasm out like that if they want to eat something. They got a nice little nucleus in them like that, right? This is just one cell. Right? And you can spell amoeba a bunch of ways. I like to put the O-E in amoeba. Yeah. Right? That's a single-celled amoeba, but this is a multi-celled U. They're both alive. They're both cells. These cells came from pre-existing cells. Life is good. And cells are the basic building block of life. I'm going to draw a really bad cube. Oh, my art skills are so good. Okay? Basic building block of life. Now, if one were thinking about a test, how many marks could this add up to? Well, one there, one there, one there. And then examples. Everybody forgets examples. You know, this example, this example, this example. So yes, you want to keep that in mind, because sometimes you'll say this is a thing, and then it says provide an example, and you're like, oh my God, I didn't study. And you're like, but it'll be fine. It will be. Always have an example for something, because that's just how the ball rolls, ladies and gentlemen. Discussion questions. Okay, so this is where you would pause the video and say, all right, identify the statements that make up the cell theory. So do you know your cell theory? Right? 3 levels. Right? With examples. More than 3 levels, kids. We're just doing the basic levels. And how did they get this far, right? And you're thinking about the microscope invention, right? Yeah, the microscope was big. So you get to thank Van Leeuwenhoek and Hooke for all their work. There's more to it than that. Now, if we get outside, this is interesting. I thought this was neat how this got thrown into BC Science 8, because they jump in with both feet on something that isn't a cell. This is not a cell. You are looking at a virus. So I'm going to put little X's wherever this thing is made out of protein. Okay? Think of it as almost like hard stuff, right? Like your nails, right? So protein. Okay? So that outside thing, right? Comes down and goes around like that. You know, it's going like this. If you actually draw the whole thing, right? Right? It's going like that. It's meeting in the middle. So you've got this protein shell, protein neck, protein tail fibers. These are all protein. It's like a protein shell. Okay. So you're like, so why are you telling me about this? This thing makes me sick. How could it possibly be alive or not? Well, then you have to take a look, and I'll label it with red. We've got this stuff right here. If we look at that stuff, well, it's blue, but I'm using red, right? You look at it, you're like, wait a minute. I know what this is. It's what Mr. Bleecker could never draw well. Okay. So it's DNA. There we go. I survived. Okay. It's some form of a genetic code. Now, in this case, this virus has DNA, deoxyribonucleic acid. So you could say, oh, so this dude and I have something in common. We both are rocking DNA as our genetic code. Okay, that's true. I guess I should stick to red. But the rest is just a delivery device. It's like the protein shell is almost like a needle for this thing to inject its DNA in. Oh, yes, it is. So you're looking at what's called the we'll call it the T4 bacteriophage. It's a kind of phage or virus. So I'm going to write that in, because another word for virus is phage. Okay. And viruses are very picky. They can only really attack one kind of host. So our friend here likes to find himself a nice big E. coli, right? And E. coli is going to be unhappy because the phage is going to be attacking it, trying to kill it. Now, that's not drawn to scale because the phage is much bigger. But this phage is very good at hooking on to its target E. coli. It can only really grab onto E. coli, and it tries to infect it. So it needs a host cell. So what you're seeing here. So viruses are basically genetic material, like DNA, inside of a container. That's not a cell. This is not a cell. And it doesn't reproduce. It doesn't follow the cell theory. So they're considered mostly to be not alive. They have some living characteristics. We'll yak, we'll talk, we'll debate. It's cool, but it has more unliving characteristics than living. Let's put it that way. So what can't they do? They can't take in nutrients, so they don't eat. Let's just say they don't eat. Let's keep it simple. They don't use energy. Okay, so it's like they don't move. They don't reproduce. So they don't even, it's like they don't what's called respire, right? Like There's no oxygen and CO2. Those don't matter to this virus. That's nothing. For you and I, it would be a big deal. For them, who cares? They don't produce waste. So let's keep it simple. No peeing, no pooping. They don't produce waste because they don't eat anything, right? They're not producing waste products. They need to get inside of a cell. They need to take them over from within. So the only purpose of life of a cell, all this dude wants to do is infect, reproduce. That's it. That's its meaning of life. Infect and reproduce, infect and reproduce. It just goes around and around and around. Because if it can make copies of itself, well, it's like it never really died. But it's not a cell, so it's not considered alive. See how it gets confusing, kids? It's fun. Not a cell, not alive. So the way viruses work is viruses have to infect a host. So they can sleep for a very long period of time. Although they don't have a metabolism, so they don't sleep. But they do nothing. But when along comes a host for them to attack, right? Go like this. Our poor little E. coli gets noticed. Right? I guess I should turn his frowny face down. There we go. All of a sudden, it's like, oh, hey, there's something to attack. And viruses, because they don't have metabolism, they don't need oxygen, they don't emit carbon dioxide waste, they don't need to eat. This is supposed to be an eyebrow, by the way. It's a terrible eyebrow. I like to draw the eyebrow like you see on the rock. There we go. People's eyebrow. There it is. He's supposed to look mean. Let's give him fangs. Yeah. Oh, it's not cellular, so these aren't real components. They don't have eyes or eyebrows, but I'm just having fun. Once they notice something that they can affect, they'll go after it. But the viruses can sleep for a very long time, lying dormant. That's what I mean by sleeping, right? Like sitting on door handles, for goodness sake, or flowing around in your digestive system, right? You've got E. coli in your digestive system. It's not impossible that this guy can get in you, doesn't infect you, but then goes after E. coli. Yep, that's the thing. Yeah, that's why we have viruses. We like to clean things, wash our hands. They can exist on surfaces. They can exist, oh heavens, they can exist in a crystalline state, some of them, for very long periods of time. But they'll try to reproduce. So, I should show you this. I thought I had this available. But when you look at the T4 phage and E. coli, let's see if I can actually type properly today and go to an image, you'll kind of see what the T4 phage likes to do. So we knew that the T4 phage existed because we've seen them under an electron microscope looking like this, right? Electron microscopes don't use light. They use electrons in a big fancy series of lenses, so to speak. And we have actually seen at incredible magnifications the attack, the assault going on E. coli. The things that look like pills are E. coli, most likely in this case, and those are the bacteriophages. The phages are attacking. They have just enough stolen energy to inject their DNA to take over our poor little friend and reproduce using our little friend, because that's what they do. Phages they're not there to hurt you. They have chosen a different victim in life, and it is not you. Some fluorescence microscopy, look at that. You can actually see how they are injecting their insidious DNA to take over this cell, right? There's another good shot of it there. Gotta love these trips to Google Photos. You really have to. Oops. So, that's what's going on with that. Oh, I just want to shrink this down. Okay. So when we looked at these phages, we were looking at them with something called an electron microscope. So it's kind of fancy. It fires little bits of electricity that are focused by magnets on the sample, which is kind of in the middle. And the image on the other side is shown on like a TV screen, so we can see what's there. So if the electrons go right through, it's light colored. And if the electrons hit solid things like the virus body, it'll show up. It's very fancy. I always wanted to play with one of these and never got a chance. Maybe some professor will show mercy upon my soul one day and say, Mr. Bleecker, come on up. Let's look at viruses under an electron microscope. I would love it. Because in your undergrad, you don't get to do that, your bachelor's degree. That wasn't something I got to do. But I did like my degree. It was very good, especially the microbiology portion. Gave me my best skills. Okay. Studying germs and viruses. So viruses will take over our poor little host and use it to replicate. Now, there are lots of kind of viruses, right? You have to consider that there are DNA viruses, so I should draw that. Right? There are DNA viruses, right? They use this kind of a genetic code. Looks like a double twisty ladder. Some viruses have a single-stranded one. Kind of goes like this. I'll draw the little, the ladder like that. So this is what DNA looks like, right? That's what we have. Some viruses have that, like polio. And then like influenza, it's rocking this one, which is RNA. It's a little bit older, and it's single-stranded. So it warrants sort of a look. When you look at DNA versus RNA, right? Here's a nicer look at it, right? DNA on the left, right? Very tough molecule. Could hang around for a long period of time, like in the permafrost inside the tooth of a poor frozen baby mastodon. Still around, like thousands of years later. And then ribonucleic acid, it's not deoxy on the right, it's ribonucleic acid. And that is a single-stranded molecule. So that's actually older, much older version. But they can both be used as genetic material. It's pretty cool. So when you look at viruses, some are like DNA, so like the phage that we were just looking at attacking E. coli, and RNA below, that would be like the HIV virus, the one that can cause the disease AIDS, the acquired immunodeficiency, right? This one here, right? That's what HIV is using, whereas the one we were looking at the above example is rocking that one. Viruses are kind of interesting. They threw this into your notes, and I thought it was neat. It's in your book. Some of these viruses are big. It's weird, because if we're trying to think, did viruses, is this like the chicken or the egg debate? You know that debate, what came first, the chicken or the egg? And we're like, well, were viruses there before cells? Huh? Or did they just show up with cells? Did they show up at the same time? These guys are big. These are really big viruses. These viruses are between 6 and 800, well, 600, that's what I meant, 600 and 800 nanometers in size. That's big for a virus. That's really big. Most viruses are, you're talking like just a few nanometers. These things are like 600 to 800 nanometers. So the Mimi virus on the top, this is Mimi here, right? And the megavirus on the bottom, they're very big viruses. And they've got they've got more genes than we expected them to have. Viruses they have very minimal software, so to speak. Not a lot of DNA, but these viruses, they have a lot more DNA. And they do some things that viruses don't normally do, right? The one on top can make little protein chunks, so to speak. I'm going to keep it simple like that. Viruses don't make anything like that. They really don't. They just use us to reproduce. So it's kind of odd. So this is about the time I was in university, around this time. Yes, I'm that old. Discovering these large viruses. And they were making stuff that never been seen before in viruses, like the little protein chunks called amino acids. We're like, huh? Viruses don't make anything. They just reproduce. So we're like, huh? Amino acid production, what's going on? Because that's something cells do. I'll just put protein to keep it simple for you, right? So they can kind of do this sort of protein subunit thing. And then along comes the megavirus, and it's got lots of DNA. It's got extra genes, right? Which is odd. It's like, why does it have so much genetic material? Like, That's odd. So could they have a common ancestor that could also make its own proteins? Because, what's going on? And then in 2015, it found some things that cells and viruses do that are the same, right? So when you make a protein, that's what you're seeing over here. So I'm going to make this kind of simple. Proteins start out, see if my grade 12s are paying attention. They start out as a straight chain like this, but then they start to fold up, right? So they'll start to fold like this. And they start to crumple on each other over time. They'll fold and they'll make these arrangements, and then you got a tangle like this. So when we look at the folding, right, over here, we're like, okay, so folded protein, that's something that cells do. Like this is how we make insulin, kids. Like we do this. But if we find viruses, right, protein folds, right, that are shared between viruses and cells, it's like, what? Huh? These are common properties. So the thing is, if they have the same properties, it sort of indicates ancestry, right? It's like some kind of common ancestor could do this. Like where did this come from? So maybe cells and viruses could have something in common. That's why science is so cool, because we got to figure out what's going on. If you look at viruses, you sort of think, are they living or non-living? They're not cells. So by the definition of the cell theory, not alive, right? Not cells, not alive. But then it gets kind of funky because you're finding these weird cell attributes they have, right? They indirectly reproduce, right? Using cells like surrogate moms. That's kind of interesting, right? They do that. They do respond to their prey by infecting. So doing nothing all the time, they can go after things they want to infect. Like, example, E. coli. E. coli got infected somehow. How'd that happen? That's a thing. So we start to look at viruses. It's like, okay, they've got some characteristics we can't ignore. But it really comes back to biggies, right? They are not cells. They are not alive. By definition, they don't create their own babies. Okay, but I, on the other hand, could say, well, they have genetic material inside, right? They have genetic material in there. We have genetic material. That indicates there's something common in our ancestry. If you want to look at something really neat, where is it? Influenza. I wanted to show you. When you look at influenza, when you look at viruses, they're not cells. They're too geometric shaped to be alive, right? So neat sort of photo. You sort of look at this. It's like, what is that? And it's like, it's this protein envelope over genetic material, right? You're like, it's tiny, right? What are we looking at? And looking at influenza under an electron microscope, they kind of look like these sort of weird pine cone things, right? And this is like mega million times of magnification. Like our light microscopes in here can go to 1,000 times. They can't touch this. You couldn't see this, right? Without something like an electron microscope, right? They're tiny and they've got crazy life cycles. They need us to divide and to reproduce. And they're just somewhere in between life and non-life, right? Are they undead? I don't know what to call them. It's still a debate. But we do know that these viruses can cause a lot of illness, right? This poor fish right here, right? These lesions on the skin, those are viral lesions. So if I zoom in on it, this is not healthy, right? You can get viral infections. And even in the ocean. It's not like our poor friend here is going to go to the hospital for medication, right? The immune system will have to clear this. And you have to consider some important questions like, should you put these critters in like a fish farm? Like you sort of have to consider that, right? When you bring the fish close together, they're going to get diseases, right? So fish farms, and you think to yourself, Okay. Oops. You think to yourself, don't use a highlighter, right? Fish farms, a bit of an issue, right? Could it cause infection? So, yeah, salmon farming is big in the province. Out in the ocean, this fish would have the ability to roam. Would it have gotten infected? Would it have infected the whole population? The disease is more spread out. It raises questions. But what it does tell you is that there are critters, on our planet that kind of make it interesting in terms of cell theory. And viruses, holy smokes, backing up, right? When you look at viruses needing to make little baby viruses, needing cells to do it, that's kind of crazy. But that's a great thing about microbiology. If you like this, you'll love microbiology. So stay at it. Okay, folks, thanks for listening. Mr. B out.